So, diencephalon is an important part of the brain which is developing in the tip of the neural tube with the next part, which is called telencephalon. Diencephalon and telencephalon are originally created from the prosencephalon. Prosencephalon, it means it is a tip of the neural tube, head tip of the neural tube. So we can distinguish here two parts, as I have mentioned, the telencephalon, but now we will concern to the diencephalon. This is an embryonic schema where the yellow color is showing the part which is called diencephalon. Diencephalon is developing along or around the structure which is called third ventricle. Originally it is a tube, but around the third ventricle, which has finally the the long diameter and very short lateral to lateral diameter. And from structures which are called diencephalon, we can distinguish the several parts. Because the original embryo or original organism is long, like a fish, for example, or primitive amphibia, then we have a part which is more ventral and more dorsal. From this ventral part and part of the pouch of the third ventricle, the ventral part is developing in a very important structure called hypothalamus. Hypothalamus is a very important structure which is coordinating the reaction of the organism to the environment and has a very important influence in the inner organs according to the production of hormones and production of some releasing factors for pituitary gland. So this is a hypothalamus. From dorsal part is developing thalamus. So thalamus is a very important mass of a gray matter, which is the largest part of the diencephalon in a human brain, and it is important, really important thing. So laterally, which is not seen here, laterally there are pouches or sacs from which are developing eyes and a visual pathway. So this is the reason why the retina and optic nerve are a part of a central nervous system, not peripheral, and they are very different from the other cranial nerves. So neurons typically are developed around the ventricle. The same is in the telencephalon, but then they are migrating to the peripheral localizations in diencephalon where there are so-called nuclei. So nuclei, which are some accumulation of neural bodies or neuronal bodies, and we call them nuclei, like in brainstem. So the original development of the brain is seen here, and this is diencephalon originally in tubular neural system, and when the telencephalon, our great brain, is growing, then it is growing like a mushroom, and this mushroom is then finally covering from a main part the diencephalon, which is then hidden in the middle of the brain, and then we can see on the surface only a small part of the diencephalon, typically the bottom of a hypothalamus. So then, diencephalon is covered mainly, and mainly the thalamus is covered by telencephalic structures. So this is important that originally it was second part of a neural tube, but in the end it is covered by overgrowing telencephalon. So I have said that the encephalon is developing around the third ventricle. Third ventricle is a spout-like structure, and the third ventricle could be seen here. The roof of the third ventricle is covered by tissue which is producing liquor, and cerebrospinal fluid is created by the gland which is called choroid plexus. Choroid plexus is also developed in the fourth ventricle and both lateral ventricles. So this is at the roof. Then the third ventricle has some different processes. The most important is a ventral process, which has two parts. One is here, and this is a supraoptic, and this is infundibular. So something was wrong. Yeah. So this is a supraoptic and infundibular recessus. So structures which we can see here in the diencephalon on this schema. I have said that the largest part of the thalamus is a thalamus. Thalamus is a structure which is bilaterally localized. Each part has a egg shape or ovoid shape, and in between them it is a structure which is bridging both thalami, which is called adhesio interthalamic adhesion. On thalamus, we can distinguish also the stria. Stria is a string which is called stria medullaris thalami, medial thalamic stria, and from which dorsally we can distinguish structures which are covering the third ventricle from the dorsal part. There are two structures, habenula, and this structure which is a pineal gland. Pineal gland is a very important organ which could coordinate our circadian rhythms. Probably you have heard about the melatonin secretion during night, which has a very important role in coordination of our vital functions between day and night. Then when we go down, the third ventricle is continuing to the communication between fourth and third ventricle, which is called aqueductus, and then we can see here the upper part of a mesencephalon. The diencephalon then is follow here with two structures, two important structures. This is a mammillary body, corpus mammillare. Mammillary body is a very important part of the so-called limbic system, and we will follow the limbic system today also. And then This is a small structure which is also important called tubercinereum. Tubercinereum is again important structure where we will see some nuclei releasing factors needed for secretion of hormones from adenohypophysis for anterior part. Then we can see this structure. I have said that we have some infundibular recessus. So infundibulum is a conic structure on which is hanged the pituitary gland. Anteriorly is a supraoptic recessus, and in the supraoptic recessus at the ventral tip we can see chiasma opticum, which is important crossing or partially crossing of the fibers of the optic nerve, as we have mentioned last lecture. So the other parts or surroundings which are associated with structures of diencephalon are those which are seen here. And this is anteriorly. This is a preoptic area. And here structures which we will have in a description of a so-called commissural white matter. This is a commissura anterior commissure. And here the second very important structure which is covering the roof of the third ventricle with the choroid plexus, and it is called fornix. Fornix is a white matter band which has a crossing fibers, which has also long fibers, and those structures are communicating with those which are localized in temporal lobe in hippocampal formation. So now we turn to thalamus. Yeah. The part of diencephalon, I have said the largest one is the diencephalon. On previous image, we can distinguish structures which are seen from the view of third ventricle. This is from lateral view. This large part of a thalamus is covered by telencephalon. If you remember the image, how the mushroom of the telencephalon is growing or overgrowing over diencephalon, here is also the coverage of telencephalic structures. So the thalamus is almost ovoid, and we can see here from lateral part. So prominent structure which is seen here dorsally. Dorsally we have a pulvinar. Below pulvinar we can see the pineal gland. Below those structures are structures from mesencephalon. It is a quadrigeminal plate. So this is a mesencephalon. Yeah. Then this structure down is a pedunculus cerebri, also a part of mesencephalon. Cranially, we can see two important structures which are called brachia. This is a brachium colliculi inferioris, which is associated with the auditory pathway. And the second one is seen here. The second structure could be seen here. This is a corpus geniculatum laterale, lateral geniculate body, which is a part of a visual pathway. And here is the relay for the third neuron. The third neuron then is transmitting the information in a primary visual cortex in the occipital lobe. The structure in the front is a tractus opticus, which is seen up to here. There is a chiasma. The structures which are in the front. Then they are nervus opticus, and this is a tractus opticus. Optic tract, optic nerve. Below we can see the structures which are infundibulum. Here is a tuber cinereum, and the last one is a corpus mammillare. So those structures you have to be able to remember to describe on the Diencephalon from the lateral view. This next view is a view which is from the top after the Diencephalon is cut it off. So here we can see very well thalami, much more, yeah. One thalamus, the second thalamus. In between there is a bridge which is called adhesio interthalamica. Dorsally is a pulvinar, and we can see here the taenia choroidea, the line on which is hanged the third ventricular roof, on which is placed the choroid plexus. In between thalami, we can see triangular structures which are called habenula, hosting habenular nuclei. And dorsally, the last one, the impaired, is a pineal gland. And you can see again here those structures, quadrilaminal plate or tectum mesencephaly, and the other structures are part of the telencephalon. We can distinguish here other structures. This is vein. The vein is in the groove between telencephalon and diencephalon, and is called vena thalamostriata, thalamostriate vein, which is draining, as the name says, drain blood from thalamus and brain from striatum. Striatum is a part of basal ganglia, which we will have this lecture also. The last structures, this is a fornix, and here we can see the lateral ventricle, left and right lateral ventricles. In between them is a septum pellucidum. In one of next lectures we will have description of the whole system of cerebrospinal fluid circulation. There we will have those structures which are delineating also lateral ventricles. And the last part which is seen here, this large mass of white matter, is called corpus callosum, callosal body. The last view is from the roof, from the floor, and here we can distinguish those structures which are very well seen after the mesencephalon is cut it off. And here is an optic nerve. This is a chiasma. And here we have a tractus. The whole system of a visual pathway which is going to the lateral geniculate body. In the middle we have here infundibulum, and infundibulum is a process on which is hanged pituitary gland. So please distinguish pituitary and then pineal gland. Similar names, different structures, different functions, but related together because they have a very important influence each other. This structure here is called tuber cinereum, and those two structures corpora mammillaria, mammillary bodies. Mammillary bodies are paired, and they were called in 19th century and before testes cerebri, because it looks like testes. So, Diencephalon then is divided in 4 parts. The key name is a thalamus, and thalamus is the largest one. This is a thalamus. Then we have very important structures which are behind, but in the fact, in quadrupods, it's up. So then it is called epithalamus. Epithalamus is 2 important structures, pineal gland and habenula. And it is important structures. The pineal gland has circadian rhythms. What is a circadian rhythms? So rhythms through the one day, through the 24 hours, day and night typically. And here are habenula, and habenulas, they transmit olfactory sensations from brain to the brain stem. So this is epithalamus. Then we have hypothalamus. Hypothalamus is this structure, and this structure is behind chiasma opticum and in between tractus opticus, containing tuber cinereum, neurohypophysis, and mammillary body. This is a coordination of the endocrine and autonomous nervous system. This is the main function of it. And the last one, which is not seen very well here, it's here, somewhere here, hidden, and it's called subthalamus because it is below thalamus. And it contains important structures, nucleus subthalamicus, zona incerta, but in reality it contains structures which are traditionally counted into the part of the telencephalon, which is called globus pallidus. So very ancient structure of a gray matter inside the telencephalon, originally developing with subthalamic nuclei. So where we can see the diencephalic structures on MR. This is MR in the transversal section and a coronary view. On coronary view, we can see here, this is a hypothalamus. And here, we can see thalamus. And dorsally, this is the epithalamus. When we look in a subthalamus is a gray matter zone which is localized here. This is a subthalamus. And now you can see this is a coronary view. Why we have something is epi, something is sub, and something is hypo. So it's a little bit dubious names, but they are seen here. So again, this is a hypothalamus. This is a optic tract. Here again, hypothalamic structures. Somewhere here we can see subthalamus. This is a quadrigeminal plate. and we go top to the top, and here is a thalamus, and dorsally is a epithalamus. So according to the parts of the encephalon, thalamus. Thalamus was originally found like a site which is typically gate to mining, gate to the mind. Why? Because almost all sensoric information is relayed here, except olfactory. So thalamus is mainly formed by the gray matter, and on the anterior pole we have a anterior tubercle, tuberculum anterius thalami, and dorsally we can distinguish the part which is called pulvinar. Through the thalamus we can see some ypsilon-like structure on each side, and this ypsilon-like structure is called lamina medullaris interna, internal medullary lamina. This is splitting gray matter of the thalamus in three sectors: lateral, medial, and anterior. Separate structures are localized dorsally and inferiorly. They are called corpora geniculata. Corpus geniculatum laterale and mediale. Medial is associated with the auditory system and lateral with the visual system. And in between is the bridge which is called adhesio interthalamica, interthalamic adhesion. So the functions of the thalamus are several. The most prominent is the relay of a sensoric information into the telencephalic structures, but the other important are motor control relayed with the basal ganglia and supporting the preparation of a musculoskeletal system to the motion. And the last one is the limbic system. So limbic system is a system which is coordinating the visual and also mainly olfactory sensations and also in animals sensations from terminal nerve, with some imprints into the memory, imprints with well-being or stress, and coordinating the feeding and breathing functions. The space relations of thalamus. The medial surface of a thalamus here is facing the third ventricle. So this is a third ventricle. And this is a thalamus. Yeah. In between is seen interthalamic adhesion, and dorsolateral from the thalamus, so here, and dorsal is going, is nucleus caudatus, a structure of diencephalon, and this is a part of basal ganglia. Between thalamus and nucleus caudatus there is a stria terminalis. So this is a groove in which we can observe in a third ventricle stria terminalis. Dorsally on the surface of the thalamus is epithalamus and all those structures of epithalamus, as we said. So the relay of a thalamus with the other structures, we have two main ways. One way is ascending sensoric information from tactile or proprioception. And then the neuron, which is originally localized somewhere in tactile nuclei, so gracilis incuneatus in oblongate, or in nucleus proprius or substantia gelatinosa in spinal cord, then the neuron is related to the second one, which has then projection to the cortex, to the cerebral cortex. So they have specific projection, and those have specific functions, and then we call them specific to thalamic nuclei. Non-specific thalamic nuclei, they have projection into the brainstem, diencephalon, and also striatum. Striatum is a part of, they are basal ganglia. And then we have some integrating thalamic nuclei, which have some integration and computation role, and intrathalamic nuclei, very small group, which is localized inside the medullar lamina. The projection of thalamus, specific nuclei. Those specific nuclei are in the lateral part. So it's a ventral anterior, so ventral lateral, ventral postero, medial, and postero lateral ventral. What they have function. You can see that here is also homunculus with projections, and those ventral nuclei have projections which are typically into the brain, into the specific areas, into the motor or sensoric cortex. So they have a specific telencephalic projections, so then we call them specific thalamic nuclei. The relay of them is a relay of a sensoric information. So ventrolateral nuclei, posterolateral, it has information from lemniscus medialis, and from lemniscus medialis they obtain the information of tactile and discrimination sensation, and from spinothalamic about pain, heat, and cold. And those information are specifically related to the postcentral gyrus, where are those sensations then integrated with the other, and then they have a real impact to the motor function or to discrimination of the position of the human body parts. So posteromedial part has the same input, but from the head, from the trigeminothalamic tract. So then it has the projection to the lower part, where is a projection of a head from trigeminal. Sensations. We have heard last lecture that sensations from the skin, sensations from the proprioception of the muscles from the head and partially neck are transmitted by the trigeminus, and this projection is similar to the rest of the body. So one of the specific nuclei is a nucleus which is called nucleus anterior. Anterior nucleus is an important part of a limbic system, this emotional integration. This is anterior, and this anterior nucleus has relay to the part which is called cortical cortical limbic system, which is gyrus cinguli. Non-specific nuclei. Non-specific nuclei are those which have relay to the striatum, basal ganglia, or the structures in the brainstem or cerebellum. So they are typically relate to the putamen or caudate nucleus, which are upwards to the structures of the deep gray matter of Diencephalon, so striatum, basal ganglia, and then they have relation, important relation, with the reticular formation and cerebellar emboliform nucleus. So in the part which is called paleo and archicerebellum. So then we have very important relay of nuclei which are localized medially and laterally and also dorsally, and they are non-specific. But when we can see the relay of them, so the medial nuclei, here pinky, they have relay with the frontal lobe. And when we have something which is related to the frontal lobe, it is related to the behavior. Then those patients who have damage of frontal lobe or this medial nuclei, they could have problems which are called frontal syndrome. Frontal syndrome is improper behavior, especially to sustain hygiene and to have a normal relay with the other people, etc. So they have very important input into the frontal lobe and then for behavior. So then we have lateral nuclei, and lateral nuclei which are laterally localized, they have projection to parietal lobe. This is parietal lobe which is sensoric. And you can see here the important structure which is a dorsally from sensoric primary cortex, and there are important integration functions about the recognition of the space, recognition of your parts of the body, and also for computation, for numeric functions, for from artistic work, and etc. So, and also nuclei. This is important pulvinar, again into the non-specific area around the angular gyrus, where are association centers, and a specific nuclei which are here. So the lateral has a projection to the primary visual cortex and medial to the primary auditory cortex. So then the medial body is auditory and lateral is a visual. So it was thalamus. Thalamus is very complicated, strange, dubious structure, very hard to imagine what this structure is doing. So when you will remember that we have a very strong input into the sensoric functions, this is one important thing, the coordination of the motion in cooperation with striatum, then the projection to the limbic system from anterior part, from dorsal part to the visual and auditory pathway, and the last one from medial part to the behavior functions for frontal lobe. Epithalamus. Epithalamus is the part which is localized dorsally from the third ventricle. It's very close to quadrilaminal plate, and it is important part of the brainstem circle of the limbic system. Because of olfaction and olfactory system is the only one which has no brainstem relay, but it is a very important function which has to be transmitted, or the information has to be transmitted into the brainstem. This is a part of diencephalon. Through it, those information are going into the brainstem. Typically through the trigonum habenulare and habenular medial and lateral nuclei. They have afferent connections, from which the most important is olfactory areas of paleocortex and piriform area, from primary olfactory cortical region and association paths of olfactory cortex, and also from those which are also archaic, also very important for autonomous and endocrinal functions, which are archicortex, hippocampus, and then hypothalamus. Between habenula, which they have a trigonal structures, they have a in-between connection, which is called commissura habenularia, vice versa connection. So from left to right, from right to left. And also this part has afferent connections, which is interpeduncular nucleus, which is somewhere here, localized, and from reticulation formation. So they have those inputs. But they have inputs from this one, from olfactory brain to the brainstem. The next part of epithalamus is commissura posterior, small important band of white matter which is connecting left and right hemisphere, and is connecting epithalamus and rostral mesencephalon. So this is a rostral mesencephalon, here is a quadrigeminal plate. So this is a communication of the epithalamus, and this is the way how the fibers are going down and up to the mesencephalon and from mesencephalon. The last part, most prominent, most interesting, is a pineal gland. Pineal gland is a structure which is hanged on the third ventricle dorsal aspect. So this is a third ventricle. So here it is intraventricular space. Here is a subarachnoidal space, so outside the brain. Here we can see habenular commissure, and this is posterior commissure. So, important functions of the pineal gland are caused by epithelioid cells, pinealocytes, modified neurons. The pineal gland also contains fibroid astrocytes. So we can then distinguish here some important tumors, and you will hear about them in pathology, which are called pinealoblastoma. Pinealoblastoma is related to pinealocytes. It's a very aggressive malignant embryonic tumor. Or you can see here also glioblastoma, the tumor which is related to the astrocytes. Very interesting function of the structure is in primitive vertebrates. Did you hear about New Zealand Heteria? No? use your Google next after lecture. It's a very, very interesting, it is a living fossilia, which is only one representative of those vertebrates. So it's a sauropod, and it has a primitive third eye, like Buddha or some Hinduistic gods. But this is in the region of parietal part of the skull, because the third eye of a human brain is a pineal gland, because it is sensitive for some photoreception. It's a very primitive photoreceptors here, which are able to relay to changing of the light through the day. Not only there is sunlight, there is a night darkness, but also the period, the time which is daylight and when it is what is night. Why it's important? Pineal gland is obtaining information from multineuronal wave from retina, which is interrelate in hypothalamus. But it obtains those informations, and those pinealocytes, which are using serotonin to create, then using degradation, melatonin, and then finally they produce melatonin, and the levels of melatonin in the blood could maintain the biorhythms and also sexual behavior. Related to the changes of the daylight, you know that some mammals have breeding, for example, deers in the October. So it's a very important that there's a day, but also circa-annual rhythms could be guided by the pineal gland. So circadian rhythms are regulated from the anterior lobe of pituitary gland also. So it's important that corpus pineale is then cooperated with pituitary gland. So melatonin, which is created by pineal gland, is increasing the levels during darkness and drops when it is a daylight. So then it's important, also you have heard probably that some people who have the problems with sleeping, they have an insufficient level of melatonin because they are working at night, they are viewing on the screens, they have a light on, etc. And also this is the problem when you have the transcontinental flight, and now you go to Sydney and then you have a completely changed your biorhythms, because the pineal gland is not able to produce sufficient melatonin also. Interesting anatomic thing is that inside there are some calcifications which could be felt by the palpation, but also seen on a CT, because this is something like a sand, and it's called acervus cerebri, so it is a brain sand. Interesting is that this gland has an important inhibitory function for other endocrine glands. Why? Because when melatonin is increasing and the activity of a pituitary gland is increasing, then almost all other hormones are in the lower part of their production curve. For example, it is clear the thyroid gland is much more needed, and the production of the thyroid hormones are much more needed during daylight, the same cortical hormones of adrenal glands, etc. And now we turn to the most important part of the coordination between neural and endocrine system, and it is a hypothalamus. Hypothalamus is a structure which is seen around the third ventricle. Here is a third ventricle, and it's a gray matter. So it contains nuclei, white matter fibers. A part of it is a part of a fornix, white matter band, and also fascicle, which is communication between mammillary body and the thalamus. And we can distinguish the thalamus in two parts from the frontal view, which is medial, which is relatively low, localized, and lateral, which is more lateral. So we can then distinguish three from anterior to posterior, three groups of nuclei. So those three, which are here green, are anterior. Three anterior nuclei are preoptic, which is important for the regulation of body temperature, and two other for you probably known from your high school, supraoptic and paraventricular, because those nuclei are able to produce some peptide hormones, which are called antidiuretic hormone or vasopressin, and oxytocin, both of them. Those nuclei have an important function for food intake and regulation of a food intake, and also the injury of medial part, so typically supraoptic, they could have obesity. And when the lateral part, so paraventricular, the injury of it, those patients have anorexia. Because of important influence of the food intake, or related to the food intake, they have very strong input into the activation of a parasympathicus. Because parasympathicus has interesting and important functions which are guided with parasympathicus. The motion of the smooth muscles and secretion of a salivary glands, pancreas, etc. Did you see the hysteria? Yeah, it's not in a Google. You have seen it. Yeah, hysteria is like this, quite very long tail, and here he has the third eye. Here, two eyes and the third. So find it. It's interesting. Hysteria novozelandica. So middle layer of the nuclei, they are important for maintain the sympathicus. So you can see that there is a space, sympathicus and parasympathicus side by side. They have interesting, important third part, which are localized in a region of tubercinereum, and those all nuclei have some relay to the important neurohypophysis. Those nuclei, please remember, nuclei tuberalia. So tuberal nuclei, they are producing important releasing factors, which are then with the blood taken and transmitted into the adenohypophysis. And last part, dorsal. This is nucleus dorsalis, dorsal nucleus, or nucleus posterior. And the second, two mammillary bodies. Mammillary bodies are interesting, important. They have input into the sweating glands, and also when they have some problems, they are injured, those patients could have hypothermia. Why? Because they have no influence to sweating, so they are not cooling the body. And it is important that also corpora mammillaria are important part of the limbic system, and probably you know that people who are fearing or when they have some emotional excitation, they are sweating. Because those mammillary body, they are guiding sweating, and also they have a relation with the limbic system. So it's interesting that from our function sometimes you have relay in a neural structures, and then we can then explain those related things. Because of hypothalamus is a close together related to the hypophysis or pituitary gland. So we're talking about hypothalamo-hypophyseal system. It is part hypothalamus. Hypothalamus is a part of a pituitary gland, and hypophysis, pituitary gland, is the whole gland, but has two parts. The anterior part is a lobus anterior, or adenohypophysis, and then the second part, posterior lobe, is a neurohypophysis. Hypothalamus has important input into the function of adenohypophysis, because here from tuberal nuclei are releasing factors, then they are with the blood transmitted to the adenohypophysis. And hypothalamus is forming corticotropin releasing hormone or factor, which is guiding the secretion of adenocorticotropic hormone. Then very similar TRH, thyrotropin releasing hormone, and gonadotropin releasing hormones. They have this very similar influence to TSH, FSH, and LH. So thyro-stimulating hormone, then folliculo-stimulating hormone, and luteinization hormone. So hypothalamus then is integrating the emotional and exteroreceptive stimuli, emotional from limbic system, and because of a very close relation to the optic pathway, the stimuli mainly from visual system, and then they are able to transmit them to the production of hormones and then guiding the functions of inner organs or metabolism or other things, heartbeat and blood pressure, etc., etc. So hypophysis, pituitary gland, is a key organ which is guiding the production of other hormones. So it's some kind of orchestration of the symphony of hormones in our human body. We can distinguish infundibulum. This is infundibulum. This is a stalk, which is a part of infundibulum. And then we can distinguish the structures which are related to this is a posterior lobe, hypophysis, and anterior lobe. So this is infundibulum, anterior lobe, and arterial lobe has pars distalis, intermedia, and tuberalis. You can see on the histologic image that those structures are neural. They are made from white matter bands. White matter structures are axons. And here is a glandular structure. So this is something which is made from stomodeum and also from so-called Radke pouch. So this is made from primitive GIT, and this is from neuroectoderma. Important different origin and important different functions, but very close together cooperation. In hypophysis, in posterior lobe, we can observe that they are productions of hormones. This is a body, and body of a neuron is localized in hypothalamus. It could be supraoptic nucleus, or it could be tuberal nucleus. And then they are producing some peptide, and the peptide is made in rough endoplasmatic reticulum, and then due to axonal transportation, they are forming so-called herring bodies, which are pouched with a high concentration of this product, and then at the tip of the axon, which is localized, where this is localized in a tuberal part of the pituitary gland, or this in neurohypophysis, and then is released, and those peptides are taken by the blood and transported to the site where it is their effect. How it's working at the anterior lobe. At the anterior lobe, the production of those releasing factors is in tuberal nuclei. Then they secreted those peptides, so adenocorticotropic releasing factor, so and then this hormone is then flowing into the adenohypophysis. And here are then the hormones which are effective in the peripheral blood, in the peripheral organs, secreted. So we have two groups of hormones, neuropeptides and glycoproteins. Neuropeptides, they have sometimes important also neurotransmitter functions. They are mainly somatotropin and prolactin, two hormones, growing hormone and prolactin, guiding the secretion of a milk in the breast. Glycoprotein, they are stimulating hormones which are stimulating the other glands. Adenocorticotropin or adenocorticotropic hormone is stimulating adrenal cortex. Thyrotropin, thyroid gland. follicular-stimulating hormone, different function in male and female. In female, stimulating ovarian follicles to product estrogens, and certainly cells in male to perform spermatogenesis. Luteinization hormone then is stimulating in a female the production of progesterone from corpus luteum, then luteinizating hormone, and in Leydig cells, this hormone in male stimulating production of testosterone. So this is the production of hormones of adenohypophysis. Adenohypophysis is localized in Turcic and here you can see the relation and two pathologies. This is a tumor which is production prolactin, so prolactinoma is a very important tumor which is causing the secretion of milk outside the breastfeeding period, so it could be also in a male. It could be in a female, and it's called then galactorrhea. So milk is, for example, produced in a female 45 years old. And this is a so-called macroadenoma. And it is enlarging this space, and it has a very important impact into the chiasma opticum because it's very near. And those patients, they could have probably no hormone functions. They are silent from endocrinological point of view, but could cause bitemporal impairment of the visual system, of the vision. Neurohypophysis. Neurohypophysis is a little bit different. It obtains the axons, which are the axons of neurons in supraoptic and paraventricular nuclei, and this is really a process of hypothalamus. It is a part of circumventricular organs, and they are producing vasopressin, so antidiuretic hormone, which has important influence into the resorption of the water and the distal tubule of glomerulus. What is important? This is important in concentration of urine, and the impairment of the function of this could cause diabetes insipidus. So those patients need to drink liters of water a day to sustain the water level in the human body. Then it's important when we have those patients, they typically have some affection in a stalk of the hypophysis. So then we can see some diseases which are called histiocytosis. So they could be different in the children, different in the adult. The second production is oxytocin, and oxytocin is a hormone mainly known from the impact during deliverance. So because this hormone is stimulating contraction of uterine muscles during deliverance. And the second thing is also the impact of the smooth muscles of the ducts of the breast. And this is very important for ejection of a milk during breastfeeding. So why those hormones could be transmitted into the blood? We have a very special system of a blood supply in adenohypophysis, and also system which is separated in a neurohypophysis. We have two pituitary arteries, so hypophyseal superior and inferior artery, and hypophyseal superior artery. They are arising from internal carotid, the inferior from petrosal, and the superior from the bear in the liquor, and in the cavernous sinus part. Those arteries are supplying plexuses. We have 3 pituitary plexuses: hypophyseal superior, hypophyseal anterior plexus, and hypophyseal posterior plexus. The anterior plexus is connected with the superior plexus by the venous structures called porta, so gate. So what you can imagine, here we have a tuberal nuclei releasing hormones. Releasing hormones are then released to the blood in superior hypophyseal plexus, and the hormone then is transmitted to the anterior plexus, and this anterior plexus then is releasing those factors to the pituitary gland cells, and then they are producing, for example, ACTH. And then this hormone is released. So this is a cascade which is used in production of adenohypophysis hormones. The separate is the production of these hormones, oxytocin and vasopressin, and they are created in here in cytoplasmatic reticulum, and then it is externally transported into the neurohypophysis, where it's taken by the posterior plexus, this hormone, and transported to kidneys, to breast, to uterus. Those hypophyseal veins are drained to dural sinuses, typically in cavernous sinus or into the petrosal sinus. So then we can, in radiology, take a sample for blood from those sinuses to estimate if there is or there is not overproduction of some hormones. So now we turn to the most strange structure of the diencephalon, which is called subthalamus. This is a section, I know that is not too easy to understand to it. Here we have a thalamus. This is a third ventricle. Here we have a cortex. And here we have a basal ganglia. Roughly, because this is a claustrum, part of basal ganglia. And we can see here the 3 parts. This is a putamen, and here we have parts which are called globus pallidus, and this is internal and external. We can see here between thalamus and basal ganglia structures which are seen here. Those structures are nucleus subthalamicus. This is nucleus subthalamicus, Louisi. And the second part is a zona incerta. What is incertus. Unknown. Yeah? Mater certa est, pater incertus est. Yeah? So, it's not to insert, because we know now some functions of zona incerta. Zona incerta is now known as a key point for motivation. So, motivation to teach you and motivation of you to learn. But also it's a motivation to eat something, to hunt something, to breed in more primitive organisms. And also the second related thing is a fear. But important to learn to be feared. So then, it's an important thing for defensive reflexes, defensive behavior. Yeah? But in 19th and mainly a very large part of the 20th century, it was an unknown function of the structure. The structure which is known from its functions is a nucleus subthalamicus. Nucleus subthalamicus is splitted from zona incerta. It is a separate nest of neurons, and it has a very important connections. The projection neurons here, they are able, like seen here, and the relations are very important of this. So, from subthalamic, you have a projection into the basal ganglia, into this part, into the globus pallidus. It has stimulating glutamic relation, but it has also connection, very important connection from basal ganglia. And this relation from basal ganglia is mediated by GABA, gamma aminobutyric acid, which has a minus. It has a plus and a minus. And you can see that it's related with basal ganglia directly in both ways. And it's important that it is incorporated into the indirect pathway of basal ganglia. This close functional relation with globus pallidus is due to because they are developing in Diencephalon. Then, during overgrowing of this band, is splitted each other. But it's secondary. So, please now remember subthalamic nucleus, and we will use it in basal ganglia and in basal ganglia circle. So basal ganglia in general, they are nuclei of a gray matter which are localized like in brainstem, like in spinal cord, inside the white matter. They are not on the surface. Basal ganglia, this is a section through the brain in a frontal orientation. Basal ganglia are those structures. They have an ovoid part which is called striatum, and corpus striatum has putamen, caudate nucleus, And then we have a part which is outside, not seen here. This is a claustrum. So this is a putamen, and here is caudate nucleus. The last part of basal ganglia, the archaic part, is the amygdala. Then we also could distinguish the older and newer part. Amygdala is something which is outside. We can distinguish, like in a cerebellum, something which is called neo, something which is called paleo. The paleo is the archaic part, and this part which is paleostriatum is now localized like to be in Diencephalon, but it's a really part of developing Diencephalon. So it has diencephalic origin. The second is called neostriatum. Neostriatum is a putamen and nucleus caudatus, and they are real part of Diencephalon. The rest, amygdaloid complex, could be also sometimes known like archy. But it is not striated, not stripped, so then it is not used striatum, but it is archy part of this basal ganglia. So, like in a cerebellum, we have a very, very, very old paleo, old archy, and new neo. Important is also the blood supply, arterial supply, and this arterial supply is then signing also this different development of those. Important is that the thalamus is typically supplied by posterior artery, vertebral or vertebral-basal system. The real neostriatum is supplied by central anterior arteries, which are part of arteria cerebri media. And the rest part, this subthalamus part, and basal ganglia, which are called pallidum, globus pallidus, are supplied by arteria choroidea anterior, anterior choroidal artery, which is arising directly from internal carotid, and supplying not only this, but also a part of choroid plexus. So then striatum. What is striatum? Striatum is a noun typically, and when in general it is used, typically it means neostriatum. And also it is caudate, nucleus, and putamen. They have a different names, different localization, but very similar structure and very similar relay. They are highly cellular, huge vascular density, and they typically produce GABA. So what is GABA? GABA, gamma aminobutyric acid, main inhibitory neurotransmitter in the brain. So then you can derive that they have important inhibition function for the structures which them they are related. They use also other, but important, we have afferent connections. Afferent connections to striatum is from cortex. From different part of the cortex, they have relay, stimulating relay from cortex. They have also important relay with thalamus. Those are stimulating, glutaminergic, stimulating relate. And then, very important, from substantia nigra with, dopamine. And dopamine is a key substance which has to be known, because this is important substance which maintains the function of a striatum. Without dopamine, the function of a striatum is not working, and then it could not inhibit the related structures. So, efferent connections. The efferent connections of a striatum, they have 3 ways. One is called direct, the second indirect, and one is also hyperdirect. So hyperdirect is not seen here. So the relay of a putamen here is to the globus pallidus lateral, globus pallidus internal, and then it has also the relay to substantia nigra. And here is important subthalamic nucleus. Subthalamic nucleus is not directly hit by these connections, but we have direct and indirect pathway. The direct pathway is to the globus pallidus, and indirect is over subthalamic nucleus. We will have once more these relations in the impairment. So, striatum. Connections of striatum are typically in ipsilateral neocortex, only in ipsilateral. They have relations according to the position of the basal ganglia. So you can observe that anteriorly they have connections with orbitofrontal cortex. Then also from the regions where is motor centers. So this is a dorsolateral frontal cortex. This is here. From parietal lobe, this is to the corpus nucleus caudati. And temporal occipital lobes, they have connections with the cauda, and they have related connections here. So there's a temporal, occipital, parieto-occipital, parietal, fronto-dorsal, fronto-lateral, and orbitofrontal cortex. So putamen has connections then with somatosensoric and motoric ways, and this is very important because lower part has a lateral and upper part has a medial connections. So contralateral projections are minor, and only inferior putamen and cauda of the caudate has a connection with contralateral hemisphere. So here, aminergic. So they have aminergic connections. Amines, biogeny means, are neurotransmitters. Dopamine is a key product. And this is the way from substantia nigra. From substantia nigra, those fibers are able to maintain the function of a putamen. But they have also some connections using noradrenaline and serotonin from locus ceruleus and reticular formation. And then it's important that the striatum has a connections related to the localization, to the functions of a limbic system, and also with the motor and sensomotor system. The real relay of a striatum is seen here. Also complicated schema. This is a striatum, caudate, putamen. And yellow one are glutamergic or glutamate using connections. And here you can see a relay to putamen. So it is exciting. But the putamen then is connected with inhibitory system. And this inhibitory system is then inhibiting the structures of the Diencephalon. This is a way which is very important. This is a direct way. The indirect way or pathway is seen here. It is a connection with external pallidum, interconnection to the second inhibitory. And this inhibitory pathway is decreasing the function of subthalamic nucleus. And then subthalamic nucleus has decreased function, so then it's not able to much stimulating backwards the basal ganglia. So then this is not overstimulating thalamus. The role of dopamine is this one. This is a way of dopamine. So what is seen here? Dopamine is needed to maintain this function of a striatum, and when this connection is lost or the production of dopamine is lost or the receptors on striatum is lost, the dopamine function is decreasing, and then it is decreasing the inhibitory function, and when it's decreasing inhibitory function, then is increased the stimulation from thalamus to cortex, and then it is increasing the function of basal ganglia, and then the patient has stiff muscles and a problem with motions typical for Parkinson's disease. So this is a very complicated multiple circulating system of inhibitory functions, and when they are disrupted somewhere, then it overstimulating the stiffness, and in fact the patient is not able to move properly. So it is a very complicated to understand to it. It's to read it multiple times. So look at this picture, which is crucial to understand how when somewhere is broken this circular or chain of circles, so then final patient has Parkinson's disease. So then, in general, we have a direct pathway and indirect pathway. The direct pathway is directly to the striatum, and then it is directly the striatum is decreasing the function of the thalamus and decreasing function of the cortex. The indirect pathway is interrelate through the subthalamic diencephalic structure, and this structure is then stimulating the system. When is disrupted also this system, which is stimulating inhibition of a thalamus. So when is not stimulating inhibition, then it is thalamus able to have an increased activity. And this is also a hyperdirect pathway from cortex directly to the subthalamic region. So this is also control or follow-up from the cortex to the subthalamic part. Basal ganglia and function disorders. Remember that they are typically movement disorders. Those movement disorders are part of some degenerative neurodegenerative diseases. You know probably very well Alzheimer's disease, which is behavioral one. And those are motion disorders. Motion disorders could be divided into Parkinsonian, which are enabled to initiate and execute motion because the most prominent stiffness of muscles. And also, when they are involved, basal ganglia in the part of caudate, head of caudate, there is inability to prevent involuntary motions. Again, impairment of inhibitory function, and it's called chorea major, or Huntington disease. And also because they have some influence into the mesencephalic motor system for sight fixation. So they patients could have progressive supranuclear palsy, which is a third one. And those patients are not able to fix your vision properly where you have to see. So basal ganglia, typically Parkinsonian disease, is related to the problem of degeneration of dopaminergic neurons or decreased density of receptors. And then the patient has increased activity of indirect pathway and decreased activity of a direct pathway. So this is finally decreasing of inhibition of subthalamicus nucleus. So then subthalamic itself is increasing the function. Now it is not well working, so then is increased activity of motor nuclei in thalamus, and finally it is increased cortical activity with following hypertonus and dyskinesia, typical for Parkinsonian disease. Really hard to understand. So now we turn to next part, last part. So description of a head in the part of the Diencephalon. So we can distinguish in Diencephalon following structures. Following structures are lobi, lobes, sulci, grooves, and then we using prominent two of them, which is sulcus centralis and sulcus lateralis, central lateral groove. We can divide the brain to the frontal, parietal, and temporal lobe. And a smaller sulcus parieto-occipitalis, and here sulcus preoccipitalis, dividing occipital lobe, and this point and pole or occipital lobe dividing then occipital, parietal and temporal lobes. So we have lobes. Lobus frontalis, parietalis, temporalis, occipitalis. So 4 lobes seen from lateral part. Cortex cerebri. Cortex cerebri could be divided into the 3 architectonic main groups: allocortex, isocortex, and mesocortex. Mesocortex is something between allo and isocortex. Allocortex has 3 layers, isocortex 6 layers. Isocortex is what is overgrowing in mammals and very in humanoid primates and most in human. This is this mushroom which is overgrowing, Diencephalon. So then it is a 95.5% of the whole surface of the brain of the telencephalon. The archaic parts, paleopallium or paleocortex and archipallium, they have only 3 layers, and the paleopallium is originally olfactory brain developing in all terrestrial quadrupods, from amphibia to sauria to birds. And now in our brain it is not rudimental, but it's overgrowing the volume of the telencephalon part neocerebral. Archipallium is something which is related to this, and archipallium and archicortex, it takes about 3.5% of all the surface of the gray matter, and it is a very important part of a limbic system which is called hippocampus, gyrus dentatus, and subiculum, the old parts of a limbic system. So, where you can see the neo. Neo is almost everywhere on the lateral and dorsal surface. The paleo is somewhere here. This is a paleo. And here we can distinguish a part which is archi, in the medial part, on the temporal lobe. So olfactory brain and the rhinencephalon. Olfactory brain is the original, which is made for analysis of distant chemical signals. It contains olfactory nerve, which is in intracranial space, bulbus, tractus, and stria olfactoria. The stria olfactoria are the strips of white matter which are distributing the signals medially and laterally to the next parts. Next parts are primary olfactory cortex, which are around, and all olfactory system is a paleocortical. So primitive 3-layered system. So, rhinencephalon, especially lateral parts, have a primitive association functions, association areas. So what is association? Input is olfaction, the presence of some chemical signal. You can smell the orange, and it could be associated in a very primitive way. Yes, it is something to be eaten. Yeah, this primitive association, the original, was developing here. Then it is growing and flourishing connections with the other stimuli from the other. So this is the primitive association function. So it has some cortical and subcortical rhinencephalic relations. So cortical is called entorhinal area. Entorhinal area is archicortex, more developed, so association system, which is association system of a limbic cortex, limbic system. Then you probably like or love or adorate orange. The other hate in the orange. I don't know if someone hates oranges. But this is the system of the cortical computation, and anterior part of olfactory cortex is activated by positive stimuli. So then it is a motivation to eat something. Then it is also related to subcortical gray matter, which is amygdala. Amygdala is the part of cortex which vice versa is associated with the negative. So you can smell that the orange is not good. So if it is 2 weeks old, it's not to be eaten. So then those negative stimuli could be imprinted into the memory by amygdala. So amygdala is a very tight associated to negative stimuli, negative emotions. And it is a very close because it is localized somewhere here, an entorhinal cortex. So it's a deeply but close together. So it has also commissural connections with paleocortex and cross projections to olfactory bulb, etc., etc. So it's important that we have some crossing information from left to right, right to left. Archaeocortex. Archaeocortex is a very old part, but it's original cortex which is able to associate the emotions, associate the functions of also of the inner organs from motion and others information taken from the body. So then it is association of external internal stimuli, and we have 3 structures which are archaeocortical: subiculum, hippocampus, and gyrus dentatus. When we look at those strange images, this is cortex, and here this is a lateral ventricle. So here we have cerebrospinal fluid, and here we have cerebrospinal fluid. But this is a subarachnoidal space, and this is intraventricular, this one. And here we have something which is called choroid fissure. So, then when you see this, here we have those structures. So this is a coronary section of a temporal lobe, somewhere here, this way. And here we have a gray matter, which is forming one S and one C. The S is a part of a subiculum, and a subiculum could be seen here. Subiculum has related surface to the subarachnoidal space. So this is the upper part of a parahippocampal gyrus. It is covered by pia mater. This layer of gray matter is rolled around and forming this structure, and this structure is called hippocampus. Hippocampus is facing also cerebrospinal fluid, but facing intraventricular space. So it is covered by ependyma. So this is a gray matter, only one gray matter which is facing the intraventricular space. And the last, the C1, is a gyrus dentatus, dentate gyrus. So those are three-layered archicortex. Those archicortical parts are a part of archaeolimbic system, so part of limbic system. Different structure is a neocortex. Neocortex, almost all neocortex has isocortical structure. Isocortical structure has 6 layers, and those 6 layers are typically different with the architecture of a cells. Second and fourth layer, they have a granular cells, and they have more association. So associational functions. The layers which are called pyramidal, they are 2 again. They have pyramidal cells, and they have a prominent execution functions. So typically motor, but it could be also not only motor. So association is a sensoric. So then, those layers are different from functions, from computations, association of functions, and projection into the layers or outside to the layers. And even isocortex is on the surface of 95% of the whole brain. The regional architectural differences could be observed according to the specialized functions of the parts of the brain. So then it is different relation between those layers in motor and sensoric cortex. So then we could distinguish homotypical. Homotypical cortex has almost the same layers, the same thickness of all layers, and then we have a heterotypical. Heterotypical cortex is a specialized cortex, and we could have the 2 thresholds. On one side, the most specialized motor and most specialized sensoric cortex. The most motor cortex is a typical agranular because they are prominent pyramidal cells. So those pyramidal cells have prominent output function. So then the agranular is seen here. In those agranular, you have very extended this 3 and this 5, and very, very small, especially the layer 4. If you compare it with this second one, which is a granular, the granular has very extended this 4. If you compare it with this is very, very large. And also you can compare this pyramidal with this one. So in agranular are dominant pyramids, in granular cells. So then you can derive very easily that the agranular is a motor and the granular is sensoric. If we look at the distribution of those architectural differences, then you can see that the motor cortex is localized in dorsal frontal lobe zone. This is a pre-central gyrus. And also here, where is a motor speech center. So Broca center. On the other side, the pinky are those which are sensoric, olfaction, auditory, visual, and somatosensoric. So they take the information, and the granular cells, which are like stars, stellar, they have a multiple connections, and those connections are distributing the signal, computing signal, analyze signals, and then they have related to those executional cortex. According to the differences in cortex, they are Brodmann areas. Brodmann areas are architectonic areas with the same architecture. We can distinguish more than 52 regions, but for our purposes to know is important to know first, two, or second, third, fourth. 1, 2, 3 is a sensoric, somatosensoric area. Then it's important 17. It's a visual. Then 44, partially 45, mainly 44, motor speech center. And then the 4, which is a motor. So you can easily to remember 4 and 44. So those are important areas where we have a same architecture. Why it's important? There are atlases of a brain which are made from thousands of not biopsies, of necropsies from dead people. And those plants are used in stereotactic surgical procedures. And we can correlate the structures with the findings of MR, for example, functional MR, etc., etc. So, lobus limbicus and lobus limbicus. I have said that limbic system is very important. In the cortical part of the limbic system, we can see some structures which are around the midline. So in a gyrus cinguli, this is a main one, gyrus cinguli, and this is association with the other structures. I have mentioned that we have many structures associated with the limbic system. We will have one lecture specialized for the limbic system again, but now I have to show you again the structures of a subiculum and a hippocampus, which are key players in the imprint of a memory and in the development of many important diseases like temporal epilepsy and Alzheimer's disease. So subiculum is upper surface of parahippocampal gyrus. Parahippocampal gyrus is a gyrus which is the most medial, also called mesial, temporal. On the upper surface, somewhere here, we can see the small area called subiculum, which is important part of the hippocampal complex. This is the most medial part. The next one is a hippocampus. Hippocampus is presented on the section like cornu ammonis. This is one. This is hippocampus. This part is hippocampus. It is emerging into the white matter, and then it is facing this lateral ventricle, and this gray matter showing like the ammon horn is localized on the surface of the lateral ventricle. Anteriorly, it is like a foot of dog, and on upper ridge of a hippocampus is this horn-like structure which is called fimbria. Fimbria is white matter which is taken the axons from the hippocampus and continues then like a phalanx, the white matter communication structure. And then the phalanx, we know that is associated with the hypothalamus. So then it is able to transmit the information between the hippocampus and the hypothalamus. Be careful, hippocampus. Why? It is hippo, it's like horse in Greece, and a hypothalamus is with Y, because it is below from Greece. So, this is a hippocampus. Hippocampus is a very important, and it has an interesting layer of white matter, which is facing the basal membrane of a ependyma. And ependyma is covering it to the ventricle. So the layer between gray matter and ventricle in hippocampus is called alveus. And those fibers converging into the fimbria mentioned above. So interesting communication of hippocampal formation. So hippocampus is then connected with the other centers. This strange image, Papez circle. Hippocampal formation has neuron. The neuron is then going through the fimbria, here fornix, going through the fornix, and then finally ending in hypothalamus. Then from hypothalamus relate to the corpus mammillare. Remember, sweating, hyperthermia if it is impaired, and then with mammillothalamic tract, with the projection to the thalamus. This is a thalamus, nucleus anterior. You have heard that it is a part of the limbic system, the anterior pole of a thalamus. And here is a neuron which has relay to the cingulate gyrus. And from cingulate gyrus, the next relay, next neuron, has a back projection through the Diencephalon to the entorhinal cortex. You have heard that entorhinal cortex is the association part of a Diencephalon. And then here's a back projection to hippocampus, to hippocampal formation. So the circle is a circulation of the information. And which information we have? This is the information, the input could be to the Diencephalon, the olfaction, to the hypothalamus, what could be visual, but also it could be from other centers which are in or around hypothalamus. Thirst, heat of body, etc., etc. And then those informations are together integrated, and then they have executive functions for heart rate, for the breathing rate, for the sweating, for the contraction of vessels, or to move with gut, to move with ventricle, to produce saliva, to produce hydrochloric acid. And you could have also the inputs from the emotional sphere, from the limbic system again. So then you could have ulcers in a stomach according to you are stressed from the anatomical examination. Yeah. So this limbic system is able to transmit the informations from inner organs to your memory, to your associations, to your imagination, and also from outside. If you see the bear in the forest in Slovakia, that you have to run out. Yeah. So, why the informations are connected? We have isocortex, mesocortex, allocortex, everything else. So then neurons have to be connected. They are connected in the local area, in the gyrus, in the nucleus, but they have projections, and those projections have to be also made together with bands of fibers, and almost nerve fibers could be seen in white matter. mostly they are axons, but could be also dendrites, which could be also very, very long. They have an organization, and we can distinguish the connections of white matter, which could be association fibers, connecting frontal lobe, occipital lobe, connecting visual information, behavior, visual, big words, for example. Then we have a commissural fibers, which are connecting the association centers from left to right, comparative or not comparative, from motor cortex from left to right to coordinate the centers. And then we have a projection fibers. Projection fibers means that they are connections from telencephalon to the brainstem to spinal cord or vice versa. They are bound to the more thousands, millions of fibers, which are called then fasciculi. So, one important communication channel is a cingulum. Cingulum is a singular around the hemisphere inside, and it is going from gyrus parahippocampalis to the frontal lobe. Then we have temporoparietal intersection area, important. This region, you can see here that we have some fibers going this way, some going this way. This is the largest and longest superior longitudinal fascicle, and here are fascicles which are between temporal and occipital lobe, and this is point where are crossing multiple bands, multiple fascicles, and deep below angular gyrus we can see crossing of million fibers. I skipped one kind. So, cingulum 1, then those fascicles, like superior longitudinal, and then also important this one, the shortest one, are so-called U fibers. And U fibers are the connection between neighborhood. For example, from pre- and post-central gyrus, between motor and sensoric. So those are white matter. The large communication between two hemispheres and also between limbic system is a fornix. Fornix is a highway of information between hippocampus and subiculum, and then between hypothalamus. This is like sledge. It has connections with mammillary bodies, connections with thalamus, and the most important is that it's a really connection between hypothalamus and hippocampus. It has so-called reciprocity connections with, so reciprocity this way and backwards, with important associative centers, typically with Raphael reticular formation nuclei in midline of reticular formation. So it is a part of a commissural fibers. So commissura hippocampalis is a part of this fornix between crura fornicis. But then this is somewhere here. This is a fornical commissure, hippocampal commission. So this is a crossing fibers from left to right, hippocampal formation. Then we have the largest one, which is called corpus callosum. Corpus callosum is the largest one with millions of fibers. Then we have a primitive commissura anterior commissure, which is transmitting olfactory brain or diencephalon or those archaic informations. Then we have a posterior commissure. We have heard that this is a transmission of a signals towards the mesencephalon, and commissura habenularum, and this commissure is seen here, somewhere here, and this is connection of epithalami. So this is important, that we have some fibers which are connecting the comparative parts from left to right, and then it is important that the brain hemispheres are able to cooperate to split functions or not only split functions, coordinate functions, and also distribute the signal to both parts of the brain. So corpus callosum is, or callosa body, contains 2 million fibers. And it has parts which is rostrum, genu, trunk, or body, and dorsally splenium. Yeah, rostrum, genu, truncus, splenium. And on the other side, when we have a transversal section, we can see anteriorly V-like and dorsally U-like, V-like forceps minor, a forceps major dorsally. What is forceps? In English, pincer. Yeah. So those are radiational fibers. If you can imagine what is the function of the corpus callosum, it's a like radial. If you this is corpus callosum, and its connection to the cortical parts of all hemispheres. And those fibers are crossing from left to right, right to left. It's important because this is also the way how the tumors, typically a tumor which is called glioblastoma, could grow along those fibers and use it like a highway to be overgrow from left to right. And it's a very important dense white matter structure. Commissura anterior commissure is seen here. It is directly in the front of a fornix, directly from the part which is called column, and this fornix connects olfactory cortex which is related to the olfactory brain, which is amygdalic complex, and also parahippocampal cortices. So it's only 4 millimeter in diameter and takes about 3.5 million of fibers. So corpus callosum 200, here 3.5, so very small number of fibers. It's like a bicycle handle. It has such a, so this is a central part which is seen here, and this is a distribution like handle of the bicycle. Yeah. Last notes about projection fibers. Projection fibers are called in the Diencephalon mainly capsula interna. Interna capsula is a V-like structure which is seen here. Where is it? It's between basal ganglia, here is called nucleus lentiformis, which is putamen, and globus pallidus. And here is a caudate, and here is a thalamus. So this is a V-shaped band which has genu, anterior arm, and posterior arm. And also parts which are not seen on this section, retrolenticular, which is seen here, and sublenticular part, below putamen. Then, upwards, it's irradiated, and if you can imagine, here is a corpus callosum from which the fibers are going this way, and here is a capsula interna, and from which the waves of fibers are this way. This crossing area, it's where is a radiatio, radiation, and this radiation and crossing with corpus callosum fibers and capsula interna, it's in a part which is also known as a semi-oval centrum. So this is capsula interna. What's important in capsula interna? Important is that there is a way between thalamus and cortex upwards and backwards between cortex and the centers which are below. The blue one are ascending fibers from thalamus to the brain. So anterior limb has in the middle the ascending and descending, like in a sandwich, side by side with this. Medial part of the dorsal limb is also communication between thalamus and a cortex. And next is a band which is from genu to the posterior limb, forming descending tracts. And in the descending tracts you have here yellow. Yellow are important because they are corticonuclear. Corticonuclear are those which are in a genu, here, this one, and corticonuclear tract is a tract to the motor nuclei in a brain stem, for motor nuclei, for chewing, for mimic, for eyeball moving, for moving with the larynx, pharynx, etc. So to produce my voice now. Next three are from neck, upper limbs, trunk, and lower limbs. So they have also somatotopic organization, like everywhere, somatotopic organization. What's important, when we have an infarction of this region, the patient could have plegia, hemiplegia of a contralateral limbs. So it's a very important structure. And because the structure is typically supplied by the vessels like basal ganglia, So then, when we have an infarction here, it's very important that those patients could have plegia, but they have probably, very frequently, plegia that muscles are not stiff, and the patient is not able to walk because the lower limb is not stiff. It's not possible to do a step. When it's a lesion in cortical region, and here is a preserved function of basal ganglia, they have a stiff muscles, and those people could walk like they walk. But when you have here the lesion, so then ischemic lesion, the patient is not able to stand, not able to walk. Then it's also important that here could be some infiltration made by tumors, descending infiltration, ascending infiltration, and it's a very important part of a communication of all functions of the brain. So capsula interna is a crucial structure to be known. So it was the all.